Hello everyone. So we are
Yeah.
here today to discuss about our our at PBL question, right?
Yeah.
Yeah.
Wait, the question is the first one is about how to perform the experiments for CDNA microarray and also how to detect them, right?
Yeah.
We share screen the question.
Oh, sekejap.
Oh,
I will try.
I think my line is a little bit lagging, so you all can continue.
I think I can share screen.
Amira, you are sharing screen, right?
Ya, tapi sekejap. Line aku macam.
I think I can try, but my line is a little bit slow. And
I think Mira, I can share screen. My line is
Okay.
yeah, can.
Yes, can.
So for the first question, it's just about CDNA microarray and how we do it, right?
Yeah.
Mm. So anyone have any idea, or you want to say something?
Guys, can you hear me?
Yeah. I can.
Yeah.
We can hear you.
We can talk about the DNA microarray first. And then just state about the protocol of DNA microarray. Means like first we need to collect the sample, and then the samples generate two types, right? Healthy and infected one. And then the second step is isolation of mRNA. And then because the mRNA has a poly A tail, so the column bits with poly T tails are poly T tails employed to bind with mRNA. And then the following the extraction buffer is used to rinse the column in order to isolate mRNA from the bits. And then the third step is creation of label cDNA. And then the fourth step is hybridization. This step involves placing label cDNA onto a DNA microarray. And then lastly, about the fluorescent tags on bound cDNA are excited by a laser, and then fluorescently, or what we call label target sequence that bind to a probe generally generate a signal. And then finally, the computer stores the data and use the result instantly.
Okay.
Yeah, we can talk about the DNA microarray.
Yeah. Okay. Wait. So in the question, right? It said that how to confirm those genes that are really involved in the production of Taxol. So before we actually start the experiment, right, we should like have
Gene sequence.
gene that what?
Gene sequence. Right.
Yeah, but we need to. I think we need to compare, right? We compare
Yeah.
gene that produce Taxol and gene that does not produce Taxol.
Oh, you mean a
I think
control
lah.
our
Yeah.
making a control in our experiment, right? So I think to confirm is
Yeah.
based on the up regulation and also down regulation, right?
Maybe.
I actually forgot like how it works. Can anyone?
Yeah. Hello.
Maybe I can search it out. I think it's like red for
Yeah.
regulation and green for down regulation. So means that the green one is our side because for CDNA microarray we remove or we treat it with something, right?
Yeah. Right.
Okay, cool.
Okay.
Then how about question 2? Question 2 I think is detection method that we use to quantify expression and provide justification. Anyone have any idea?
I think I have idea about question 2. They are asking about the detection method, right? So I think my opinion is you're using the SYBR green, which is actually using fluorescent dye marker as a detection things. And I think is suitable because fluorescent dye can produce a signal that is not. I think it is indirect with the DNA synthesis process. Yeah.
Yeah, yeah, I agree. But I think
Yeah.
doctor did mention that we for group 1 and to group 3 we have to use Tagman probe. If I'm not mistaken lah.
Oh. Okay. Yeah. I
Yeah,
think.
because tak silap lah rasanya.
Mm,
Sekejap. I think
also
in
remember
WhatsApp
that.
WhatsApp orang ada cakap kot rasanya. Tak silap.
Oh yeah. Yeah. I miss.
Yeah, so I think
Sorry.
it's okay.
Okay. Okay.
So I think we can use we have to use the Tagman probe lah for the qPCR.
okay.
Then, guys, what are the difference between the real-time PCR and normal PCR? Do you guys have?
I think the results validity, reliability, like RT-PCR results is more accurate and require less time, I think, than PCR results.
Yeah. Yeah,
And
and then
then,
I think real-time PCR it focus more on the for the normal PCR, sorry, we use like we want to amplify, right? For the real-time PCR, it's more to like to quantitate the product.
Yep.
Yeah. Yeah. And I think RT-PCR can be observed while in the runtime, but the PCR can't be observed in the process.
Yeah, agree.
that QRT-PCR is better.
I think so. I think it depends on the things that we want to do with like what we want. Yeah. By the way, I think that is all right for our discussion.
Yeah.
Okay.
Then we can start do the assignment. I think we can divide it into half. The first group will do first assignment, first question. Second group will do second question.
Yeah, can.
Okay.
Yeah, I agree.
Everyone.